And that was that, in fact, CLA wasn't the only thing that had changed in our butter. We also massively increased this fatty acid, so-called C18-1 trans. That's a trans fatty acid. That's what we find in hydrogenated vegetable oil. That's what we've been told for the last 10 years is the source of all evil. It's been wiped out of our food, certainly in the United States and Canada. Restaurants aren't allowed to use hydrogenated oil in cooking, and so on and so forth. So what on earth's going on? We fed a butter which was enriched in CLA, but even more enriched in trans fat, and it actually reduced cholesterol rather than increasing it. Well, basically, it turns out that the type of trans fat that we have in milk, which got through to the butter, is very, very different to the type of trans fat that we've got in hydrogenated vegetable oil. Along the bottom axis there, these are just all different, what we call isomers. The double bond is a slightly different position in each of these isomers. And in hydrogenated fat, we have a broad range of different isomers. The red bars there showing you a broad range. In our ruminant fat, in our milk, there's one major isomer right in the middle of there, that so-called trans 11 isomer. And what seems to be emerging is this one particular trans fat might actually be good for you rather than bad, and it might be the other ones which are bad for you. Our problem now, apart from proving that is definite, is how on earth do you get the public to understand that? They've now been told year after year that trans fat is bad for you. All of a sudden the scientists have changed their mind again. Yes, it is bad for you, but not this one. So that's going to be an effort in itself. But it might be good news for the dairy industry. Perhaps we can produce a super milk, a milk which is rich in CLA and this particular trans isomer, which may actually have health benefits. So we're hoping that that will pan out in the end. Right, but I'll speed up a bit. But fish oil, as I say, that's something you'll all be familiar with. In terms of heart disease and metabolic disease, there's a significant amount of evidence, certainly, that it reduces cardiovascular disease. That started when we discovered that Eskimos had very low incidence of heart disease until they moved to Western society. And as soon as they started eating what people ate in the rest of Canada or the rest of Scandinavia, they developed the same risk. And it seemed to be the omega-3 that was actually protecting them. More recently, it looks like it may alleviate some of the symptoms of metabolic syndrome, and even more recently, there's evidence building up that it might actually prevent or reverse this buildup of fat in the liver, the non-alcoholic fatty liver disease. So, the problem with this is that the ones which actually just seem to have the benefits, are what we call the very long chain ones, with lots of carbons in them, 20 or 22 carbons, and lots of double bonds. That one's got 5 double bonds in, that's got 6 double bonds in. And we really only get them from marine fish and marine mammals. And most of us have objections to eating whales and seals and things like that. Therefore, we only get it from fish. That's because they get into the tissues of those animals because they're part of the composition of the marine algae, which many of these, which within the food chain in the sea, they ultimately end up eating. What we do eat, however, is lots of what we call a parent molecule. That's 18 carbons, 3 double bonds, so-called alpha-linolenic acid. And that exists in lots of our vegetable material, in many of our plants. If you go into a health food store, you'll see lots of supplements, for example, of flaxseed, which is essentially the same as linseed. And all animals have the ability to convert that C18-3 all the way through to these long chain ones. But we're not very good at it, and humans are particularly bad at it. So could we actually increase the amount of the alpha-linolenic acid in the diet of some of our farm animals and get them to convert some of it into EPA and DHA, and then we'll enrich our meat and our milk with that, and we'll actually benefit from it that way. Well, a very simple experiment we've started off with recently is just to take beef from animals which are fed either on grass, which most of us assume that they normally eat, or on what we call a concentrate diet. Concentrate diet are things like cereals, barley and things like that. And feeding concentrates to cows in particular is a very, very common practice in North America. Most of our cows get some grass at least some of the time, but in North America, concentrate diets are very very common. And it changes the fatty acid composition of the meat. The major fatty acids don't change very much. So you can see palmitic acid there, just that big bar just on the left, is a saturated fat. It's very rich. We've got oleic acid. There's not much different. The changes really are down at this end. And they're vanishingly small, unless you blow it up as we have here. But in fact, what we did find was if we compared the grass-fed beef with the concentrate-fed beef, that from the grass animals had significantly more linolenic acid in it, had significantly more of the EPA, that's the one with 20 carbons and 5 double bonds. It didn't actually affect the DHA, but there were changes. Now, are those changes enough to have an impact on the effect of the beef on heart disease? A different mouse model this time. This is a transgenic mouse. It contains a human gene. And that human gene is a mutant of a gene that we normally have, called ApoE, which causes people to develop hyperlipidemia, high plasma cholesterol, and have an increased risk of heart disease. And if we put this gene into these animals, they will also develop hyperlipidemia. They'll also develop atherosclerosis. They won't keel over and have a heart attack, but it's a good model. So we fed these mice our beef from either our grass or our concentrate-fed cows. And I was quite surprised because the differences in the composition were very small, but we got an effect. We didn't get an effect on plasma cholesterol, but plasma triglyceride, the other major fat in the blood, was lower in the grass-fed animals than the concentrate-fed animals. Increased triglyceride is part of the metabolic syndrome, so we got quite excited. Have we actually managed to reduce atherosclerosis just by eating beef from grass-fed animals compared to concentrate? Unfortunately not. The difference wasn't big enough. What you see on that side is a cross-section through the aorta, the major artery of the mouse. And the red bit where it says lesion, that is the start of an atherosclerotic plaque. That's the start of the clogging up of the artery. So these animals develop atherosclerosis. But the difference in the diets was not enough to actually give us a significant difference in the development of atherosclerosis. And where that work's going now is to try and further increase the N3 or even the 18.4 content of the diet of the cow to load that meat with more of these precursors with the hope that we might be able to have an effect. So that potentially, again, we could produce beef which could be advertised as being potentially healthier. Get rid of what? Ultimate transgenics. If I can persuade Keith Campbell to get involved, then perhaps we can go all the way and produce a true hybrid between our fish and our cow and produce something which is even healthier. Final thing I just want to say a little bit about is one of the major research interests of the whole of our division and beyond the division within the university. And that's so-called fetal programming, or the developmental origins of health and disease. And this all goes back to what was called the Barker hypothesis, which suggested that fetal undernutrition in middle to late gestation leads to a disproportionate growth in fetal growth and programs later coronary heart disease. And David Barker in Southampton came up with that in the mid-90s. It has its origins much earlier on, however, and the data that David Barker used to actually predict this, a lot of it was historical. And this is the one which is the nicest story in many ways, and it relates to what was called the Dutch hunger winter during the Second World War. And for a while in Holland, Germany barricaded the movement of all food into areas of Holland, and people within that area were literally starving to death. They had very little food. They were eating tulip bulbs, they were eating candle wax and all sorts of things. But it was a relatively small period of time. Then the Allied troops got in, they airlifted food in, they dropped it, and liberated them finally. During that period of the Dutch famine, there was a number of women who were obviously pregnant. And good records have been kept ever since then, all the way through to what happened to their offspring. And it was quite surprising. Those women who were very early on in their pregnancy, during the famine, their offspring had an increased rate of obesity, particularly the women. They had a more atherogenic lipoprotein profile. Their LDL cholesterol, their bad cholesterol was up, their HDL cholesterol was down, and indeed they developed more heart disease.